Started. First of all, one more time, good morning, good afternoon, good evening, depending on where you are on this planet. Thank you for inviting me to speak to you here. I entitled the talk Digital Twins in Brain Medicine. And what I had in mind is I wanted to share with you some of the developments, computational, theoretical, that we have moved forward in terms of large-scale brain modeling, and then translated into clinical applications. And in our hands today, it means applications to epilepsy, but that is really becoming wider and wider, healthy aging, pathological aging, and psychiatry, in fact. First applications in schizophrenia are emerging. But let me walk you through that and show you how it actually arises and what it looks like. Yeah. So first representation of a real world data, they are actually feeding in a digital representation that you see represented here on the right hand side. To the left, real world; on the right hand side, a digital representation. And please do not interpret a digital twin as a detailed copy of the really existing system. This is not what one typically understands under digital twins. It's more an operation cycle. It's a workflow which is actually circular and goes back and forth between the real and the virtual representation. In fact, you're feeding data from the real world into the virtual representation and integrate it into a cohesive reference system, which you then also equip with mathematical equations, so with a computational model. And think of the computational model as the totality of our knowledge we have in neuroscience, or at least a subset thereof, that we represent in a formal fashion. That is a large-scale brain network level in this particular case for the large-scale brain models. And then where you implement some of the cause-effect chains, very often also vertically from microscopic to macroscopic behavior, that you implement within this virtual brain framework in a form that you can actually run autonomous simulations. This you do. And this allows you to make predictions that then actually allow to make statements feeding back into the real world, either optimizing processes or identifying certain parameters that are relevant, such as in pathophysiological situations. We will speak about that in more detail. So that's a framework. And what I want to do is, in the first part, is I want to step through this with you and show you how we actually build such virtual brain twins. Key for that is anchoring it in the same brain reference space. And this is something that you find in EBRAINS, which is a digital neuroscience infrastructure that has been created about 2 years ago as an outcome, as a deliverable of the Human Brain Project. And that is available actually to the entire community. And actually everything that I will speak about, you will be able to access there actually freely. All you need is a user account. Yeah? So in this brain reference system, we are anchoring the information, which is data, tools, or mathematical models. Yeah? So it can be on the region level. On the region level, you can then, in particular, so you choose a parcellation in particular region, then you can represent variability and different types of data. Yeah. This can be connectivity data, or you can go all the way across different scales to the micrometer scale, where we can actually import cytoarchitectural data. But it goes also across modalities, so ranging from cells, fibers, receptors, all the way to time-resolved data, so-called functional data, where we can represent them in the same reference system and map then data distributions, as you see represented here, for instance, across the cortical depth, upon the parameters in a model. And the mathematical models are for a given scale of representation, such as point neurons or brain region models, population models. You can map the parameters directly upon the model. This can be done in the framework using Python notebooks, for instance, yeah, or if you want to parse a type of data, exactly this space here, you can actually parse. You can click on a region and you can get a menu that allows you, with a pull down menu, allows you to click the individual elements that you want to have. Yeah. So it's not automatized, yeah, but you still, of course, you need to interact, but interoperability here is a key issue. I want to focus today on the model aspects. So the level when we're talking about a large scale brain models is on the full brain network level. Yeah, key element of a network are nodes and links, and the network nodes represent typically individual brain regions, as you see represented here, and those you can represent either as a mix of, for instance, excitatory and inhibitory neurons. You can actually split it across the layers, or what we do here is typically we cluster it in using mean field theory, typically in populations, and then we reduce the equations under certain approximations. There are many examples around: Janssen, Ritt, Brunel, Wang, different types of models depending on the type of approximation you make. And then you get a low dimensional representation, which we typically call a neural mass. Yeah? So this neural mass is our network node. And then we can build a large-scale network equation that is actually connecting these different nodes. And this is representative for the mathematics that we deal with. Psi being a field, in particular a vector field for the location x, time point t. This is typically so this is a time differential, and psi being a vector field, it depends on the dimensionality that we have in the neural mass, which ranges between 2 and 12, typically, the biggest one going all the way up to 12. Yeah. Then this is a mean field. This is the mean field, so the population model, the neural mass at location x and t. Let me jump to the last term, which connects you across long distances via a connectivity skeleton, which we obtain typically from imaging data of various sources, yeah, or of an individual, which we will speak about in a moment, about how we can get this in vivo imaging for the human. Yeah, but this is the integral kernel. This is the transfer function S being translated from a location x prime and undergoing a time delay depending on the distance and the track lengths. If we go at high resolution, so inside the cortical or subcortical areas, then we have also intracortical connectivity. And the intracortical connectivity is typically translationally invariant, which translates mathematically into a simple integral kernel via convolution. So this is the mathematics that we are dealing with. Maybe one last thing I should point out, please recognize that the large-scale connectivity skeleton GXX prime is not translationally invariant. This one is translationally invariant. For the experts, the left-hand side part, all the way up to the local connectivity, can be expressed also via partial differential equations. This is impossible for a variant integration kernel because you have time delays via signal propagation there, and the lack of time invariance is actually not allowing a partial approximation. So this renders it already difficult on multiple levels, numeric implementation being one of them. Yeah? So let me summarize how we organize such a building of a virtual brain. We do MRI imaging. We extract cortical and subcortical areas and represent them in three-dimensional physical space. Then it undergoes a certain parcellation. So that gives us a network representation where we each region represent by one of these neural masses. Then for the human, typically we use diffusion tensor weighted imaging. This allows us to perform tractography and build a connectome, yeah, a connectivity matrix. This is also co-registered in the same brain reference space that I was talking about, yeah, and connecting the individual individual network nodes. And then, because we span it in three-dimensional physical space, we can actually also, in addition to this, introduce now the sensors. In epilepsy, for instance, what we do, we deal with stereotactic EEG, where we drill holes into the skull and introduce a so-called Talarak electrode that has 10 different contacts. Stereotactic because it allows us to go down inside into the volume. Then we can sample the locations inside the brain and performing a source to sensor mapping. But that, of course, goes also non-invasively, MEG, MEG, or the functional MRI. Yeah, so the source to sensor mapping is actually very important if we want to go later to the clinical translation for the individual patient. Having built this, it allows us to do something like that. Yeah, so we equip each network node with the neural masses that we spoke about, connected according to the integral differential equation that I showed you. And we can run autonomous simulations that you see represented here. So based on the a particular parameterization for a patient, we talk about this later, how we do that, can run the simulations. In blue, you see the individual network sources, nodes, these are, and then you see the activity as measured by the SEEG via the forward solution. So this is physics, mapping the sources to the sensors, and then you map it on the individual electrode contacts, and you see the time series here that the clinician would measure at the location of the patient's own implantation scheme. So this is what you see represented here. And in red, what we do there is actually we integrate the energy that there is within a small time frame as the vertical bar goes through here. So you see actually where the activity is being measured in the three-dimensional physical space. And what you see here is the onset of a seizure, how it propagates through the network, recruits other areas until it stops at the end. So this is a very classic seizure event. So here you see empirical data. This is coming from Kathy Chavon's lab. On the left-hand side, this is not SEG, but ECOG. So this is a film that is being mapped on the surface of the cortex. So you see the individual contacts here, and the time series that correspond to this activity are plotted underneath. But there where the red cross is, you find also implanted a micro array, where you actually get a high resolution collection of individual data. So you see actually the organization on two scales, on a millimeter scale, on the several centimeter scales. And you see this beautiful pattern formation, some spiral patterns here on the left-hand side on the ECOG, but also propagating activity across the cortical surface on a very detailed scale. Yeah? And this is just to illustrate to you what data they look like. And actually there is a need to go to a more microscopic scale, because on this scale, the way how we have reconstructed the twins, the virtual brain twins, the way I have told you now, it's actually too sparse, too coarse, I should say. Yeah? What it looks like is actually as follows. What I have shown you initially, typically we work with 100 to 200 network nodes. This is what you saw in the previous illustrations, which provides you with an average number of 10 to 20 square centimeters per brain region. This is huge. You will actually not be able to see this high resolution activity that we just spoke about. So for this, we need to go to higher dimensions, and we do this here by a factor of 1,000. That allows us actually now to define the intracortical connectivity. This is the local connectivity term that is translationally invariant that we discussed. Before, it was not possible because it was ill-defined. Yeah? And it gets us to a resolution of multiple millimeters, square millimeters, that allows us actually to resolve this type of spatial-temporal dynamics that we saw empirically, and allows to actually describe propagation across the cortex, yeah? And a reorganization in terms of spatial-temporal patterns. Yeah? That's one thing. It requires also high-resolution data, and here I'm providing you with an example that we have actually in EBRAINS. This is a magnificent data set that comes from Cyril Poupon at Neuroscience in Paris, who has performed high-field ex vivo imaging. So this is not something we can do in vivo, and where we can actually obtain a beautiful connectome at highest resolutions on the multiple hundreds of micrometer scale. And here you see actually a full tractogram represented at a sub-millimeter scale, and you can actually zoom into the individual areas that you see represented here, getting us to the 100 micrometer scale, providing this beautiful arborization and the streamlines that you see represented in this figure to the left. And these type of figures, the coloration is indicating typically the orientation of the fibers in three-dimensional physical space. We can take this type of fiber system, either this one or other high-resolution fiber systems, and what we do is actually we build this surface mesh, this geometry of based on the MRI, and then we actually take the connectome, you see that represented here on the left-hand side, and we interface it, or sorry, integrate it and then interface it with the tessellation of the cortical mesh and compute the intersections of the streamlines to build a connectivity matrix that you see represented here. It's huge. Left hemisphere, right hemisphere, interhemispheric fibers, cerebellum, and some subcortical areas projecting into all the other areas that you find here. This is a high-resolution connectome that we are typically then working with at this submillimetric representation. That allows us to build very beautiful, remove this, beautiful high-resolution geometry that we need to describe what I showed you empirically. Here in this example, you see, for instance, an intersection, so in this coronal slice where the cortex actually folds beautifully into the hippocampus. So here we have the different CA regions, corn ammonis 1, 2, 3, and 4 that are represented in this particular fold. On the bottom here, we represent this unfolded, mapped on the unfolded cortex and on a sphere. Yeah. And on the next slide here to the left, this is the same map, CA1, 2, and 3, 3 and 4, and the subiculum here in blue, represented on the right-hand side, and where we are actually performing a simulation. And we have increased also the degree of hyperexcitability in there. And it shows you how actually the dynamics can evolve, organize itself in different patterns, yeah, and start propagating out into the areas that have lower hyperexcitability, building a front, building these traveling wave patterns in between until the seizure actually stops towards the end. This is the type of spatiotemporal dynamics we have the capacity actually to describe, and that is actually known empirically. Yeah? Just another illustration of things that can be done now that were not possible to do before is what you see here is the rhinal cortex and the fibers within the corresponding to it in the same hemisphere, intrahemispherically, and projecting on the other hemisphere, interhemispherically. Can be characterized, of course, the connectivity using graph theoretical measures. But more importantly, in this context, we can actually, as you see here, increase the hyperexcitability in the rhinal cortex and actually then check on what happens in the individual simulations. This is what you see here. Please, notice this. The activity starts here. Now focus on this area where my mouse is, my cursor. Did you see it upcoming, the activity, but not propagating horizontally across the field? But due to the heterogeneous fiber system, the white matter fiber system emerging on the right-hand side, and only then evolving into this beautifully organized spatiotemporal pattern formation. So this is something that can come out only in a system with spatially variant connectivity. Otherwise, you would have only the individual propagation across the surface. So this type of activity at high resolutions can now be performed. Something that is of interest in the same context is also to go vertically across scales. Sometimes you want to take one area and actually describe it in detail using a cellular level representation, not the mean field approaches. And for this, we had to actually develop technologies that did not exist that allow us actually to couple different simulators. This is a nest simulator, or the neuron simulator works equally well with another simulator, in this case, a full brain network simulator called the virtual brain. And we built adapters, co-simulation technology, that allow the simultaneous use of a region of interest using a cellular level simulator here for point neurons nest, and the rest then using the mean field models that are being represented in 3D space using the virtual brain. And this works quite well. As an example, I'm showing you here some data. This is coming from Javier de Felipe's lab in Madrid, using electron microscopy, reading in the data, digitizing them and reading in the data, and represent them in the human brain atlas in the same brain reference space as the models, then representing the arborization of the dendrites and the axonal projections, and computing them as probability clouds, as you see on the right-hand side. This is coming out of Michele Miglioro's lab, yeah, and where probability clouds are being computed corresponding to individual neurons. This is being done for multiple thousands, 100,000s of neurons, and allowing to rebuild actually one of the CA regions of the hippocampus of the human. We're talking here about the human hippocampus, yeah? And representing it in three-dimensional physical space, but here now on a point neuron representation. And this can be integrated in using the co-simulation and performing simultaneous simulations of the virtual brain and that particular area. Here you see actually the CA1 region where it's on the left-hand side isolated in complete isolation for the same parameter settings, running integrated in the virtual brain, which receives via the connectome input from the outside, from the other regions, and projecting back into the other regions. Yeah? And there you have seen on the right-hand side, you actually find this beautiful pattern formation. Yeah? This here is, of course, just to show you how the technology is organized and what it does. This is not an explicit scientific study. This is a feasibility study with data available and the methods available here on Nature Computer Sciences. This is by Gandolfi et al. And the simulators are also available. So this is more proof of concept of the technical feasibility. Please check it out. Yeah. So how can we now take this horizontal, or let's say a multi-scale workflow, including some of the multi-scale elements, in order to make it usable for the clinical translation? And there are different possibilities. And I told you I will focus on the epilepsy today. Yeah. Here on the left-hand side, you see again a representation of the digital twin cycle from the human brain to the virtual brain twin that we construct using the approach that we discussed, which allows us then to make predictions. Key, what I wanted to show you is actually how we are closing this cycle, how we are actually making these predictions. So first of all, there must always be an idea about a cause effect, how you implement it. In epilepsy, so using the same equation as I showed you earlier, here the local interactions are missing, but the neural mass is here, and this is a large-scale connectome with the time delays. It's a hyperexcitability that is linked to the epileptogenicity of the network, yeah, which is our representation of the cause effect of the pathophysiology in epilepsy. We'll speak more about this in the next slides. I enter into this in much detail. In Alzheimer's disease, we can actually estimate the deposits of beta amyloid and tau, the different plaques, and to represent it in the appropriate neural mass models. In multiple sclerosis, it's the time delays. They play a particular role, representing demyelination of the time delays via fiber propagation, representing the different lesions that then can be actually estimated. And the way we use a virtual brain twin in this type of applications depends on the question that you want to ask. You can either ask it as an autonomous in silico platform for simulation. That's what I implied and showed you until now. Or we can use it as a representation, personalized or individualized to a maximum that then can be used in the context of functional brain imaging data to perform Bayesian inference, to infer parameters, those that you see represented here, that may have some diagnostic value and that then make predictions so they can actually be used in the real-world real-world applications. And that's actually what we're going to do here. And the reason for that is the following. So I'm centering now on epilepsy. In epilepsy, generally, it's 1% of the human population that is epileptic at some point in their lives. Of this 1%, 2/3 can be treated and controlled pharmaceutically, yeah? Which means 1/3 are drug resistant. Yeah? And for those, other approaches to control the epilepsy or to cure the epilepsy must be found. And to this end, technologies have been developed to explore the epilepsy better. And here you see Jean Banco and Jean Talarac in the 60s who have actually developed for clinical exploration the SEEG technology, these needles that you saw earlier being inserted into the volume of the brain. Here you see the sizes. So we are talking about the individual contacts being on the multi-millimeter scale, and then allowing us to perform this type of measurements. Typically, the patients are entering into the hospital and stay with the implantation for 2 weeks. Yeah, and then they are being monitored to register spontaneous seizures, and if not possible, then you stimulate these electrodes in order to trigger a seizure, in order to actually get this type of information about seizure propagation. What is worthwhile to point out is that over the last few decades, the surgery success rate has not improved. In fact, what we are after is to identify the focus or the epileptogenic zone that is sometimes multifocal in the three-dimensional brain network, that then is from which actually the seizure starts and then propagates and recruits other brain areas, being responsible for the emergence of the seizure. This is a focus, this is the objective of this clinical exploration via SEEG, and then for the intervention, you actually enter surgically and remove the epileptogenic zone. What you see here across different epilepsies, this is lesional temporal lobe epilepsy, non-lesional, and extratemporal lobe epilepsy. What you see is that in principle, the surgery success rate has not much changed over the past decades, across all epilepsies being around 60 to 70% success for the temporal lobe epilepsies, and dropping down to 25–30% for frontal lobe epilepsies. Yeah? So there is lots of improvement, in fact, space for improvement, I want to say. So taking the digital twin cycle or circle, yeah, this is what we do. We collect the data for the patient, we reconstruct the digital brain twin for the patient, we equip it with the appropriate mathematics in three-dimensional space. And then we are actually using, in a second step, a Bayesian inference to estimate the data. So we simulate the data and then perform inference in order to better personalize the patient brain's model, digital twin, with an identification of the epileptogenic zone in order to make a prediction and get it back to the treating clinician. This is what we do. And what I want to speak about now, enter a little more mathematical detail, is actually what the neural mass model looks like. Yeah? And what we have done, and this is very specific for epilepsy actually, is that we have built a canonical model with multiple time scales that allows us actually to map existing physiologically derived models, such as a Janssen-Ritt model, such as classic population or all population models that are available and actually anchor it in its parameter space. And what you see here is a codimension 3 model. Codimension 3 meaning that you have 3 free parameters, and this is actually the set of equations that goes along with that. Careful, this is a generic model. Yeah, this goes back to the 90s. Dumortier and others have studied this model and performed actually a systematic bifurcation analysis, yeah, by essentially unfolding it around this particular point here, which is actually the Tarkins-Bogdanov point. Yeah, so you have actually the intersection of a SNCC bifurcation and a supercritical Hopf bifurcation at this particular point here. And here you're plotting this done numerically now, the amplitude scaled from 0 to a max value between the individual bifurcations curve, and here the frequency. For the epileptor, which is one of these models that we use, which has multiple time scales, you'll see actually that it's topologically equivalent. The same applies to the Janssen-Ritt model. So we have actually taken this model, the generic model, and we have actually extended it to include a slow time scale that can also be derived, either introduced phenomenologically or derived from extracellular field effects, such as extracellular potassium that operates on a slow time scale, that provides us with a fast, slow time scale separation that allows us actually to still use this parameterization of the particular model. We can do this mathematically in more detail, as you see represented here. I told you it's a codimension 3 model with 1, mu2, mu1, and mu3 parameters that are to be unfolded. And here you see actually 2, mu1 and mu being represented here, and the Bogdanov's or Tarkins-Bogdanov point is here. Said differently, every system that has a Tarkins-Bogdanov point in its neighborhood in this unfolding has a similar parameterization as this particular model. And that allows you actually to describe changes from onset to offset, so crossing the individual bifurcation lines. I'll give you one example here. Look at this one here. This is the saddle node, saddle homoclinic bifurcation. And as you move from here, from this point to that point, you enter into an oscillation, and here you leave the particular oscillation, yeah, on the right-hand side. This is represented in this bifurcation diagram here, where you actually, this is the ictal activity, where you leave through a homoclinic bifurcation, you jump on the resting state, and then if you go down, this is what is happening on the slow time scale here. You move to the right-hand side, and through a saddle node bifurcation, you jump back onto the closed orbit here and oscillate as then the slow dynamics goes back down. This in 3D, when you unfold it, provides you with a spiral attractor, this being the resting state here. Yeah, this is how you can actually describe one set of types of bifurcation pairs for onset and offset. And you can do this actually systematically. And the way it works is you can actually, here on the left-hand side, you take this set of equations. Yeah, I remind you, they are generic, these are normal forms. You can actually take the 3 parameters and write them in 3-dimensional and spherical coordinates and reparameterize the equation system. And you can actually reduce it on this particular form here, and can actually show analytically that through these individual steps in the new coordinate system, that all the bifurcation curves are lying on the sphere. Yeah, so it doesn't change topologically. So with a 2-dimensional description on the spherical surface, we can actually describe the entire system completely. And this is a result that comes out of this. This is what you see here. The Tarkins-Bogdanov point is this point here, and these are the corresponding bifurcation curves. Yeah, and then this is the different types of seizure types that can be described and can be identified. So you have a taxonomy of different seizures. This and the map itself the different type of behaviors here is generic. Said differently, every other physiological model that has a TB point can be actually represented in here. And work in the collaboration with Bill Stacy and other colleagues, but this was led by Bill Stacy, that tested this actually empirically by looking at DC recordings, yeah, across different hospitals, yeah, across thousands of different seizures and hundreds of patients. You actually performed a sampling of this interior taxonomy and tried to reproduce the amplitude scaling and the frequency scaling to identify the different types of seizures. And here you find it for the seizure onset, which is a supercritical Hopf onset. Or here in this seizure, you see a saddle node onset. So you have a DC jump, DC shift, and then the fixed amplitude or evolution, yeah? The same thing for offsets. So they've done it very systematically, very beautifully, and identified essentially two seizure classes based on the bifurcation types, which we are actually using systematically. And the one is saddle node onset and homoclinic offset. This is the most dominant one, and the other one was linked to a Hopf bifurcation and homoclinic on and off. Yeah, and this was predicted actually when you perform a complexity analysis. So this is the same all in the eLife paper here by Marissa Saggio. You can actually perform a quantification of the onset and the offset pairs in terms of complexity, and quantify the complexity in terms of number of parameters that you need to tweak in order to drive the system towards this bifurcation pair. And in dark colors, what you see is actually the system with lowest complexity. And in terms of numbers that are inside, what you find is actually, these are the biggest numbers for the darkest colors typically, that you find most of the seizures. There was no correlation between the seizure type and any type of clinical behavior, so clinical symptoms or location. So it's really a dynamic phenomenon. So that's number one. This was a very detailed description in terms of the dynamic features of the model that we use on the network nodes. This is what we implement on this high resolution network. Now, this twin we could use now for simulations, but how can we perform inference? For inference, we need to essentially have the model to be able to simulate it, but we need to also be able to estimate the parameters, to sample the parameters. Typically, we take some Monte Carlo procedures, Monte Carlo based Markov chain approaches in order to infer the corresponding parameters. We need to make decisions about the data features. And what you see here is a seizure, pre-ictal activity, seizure onset, very fast activity, and then the amplitude increases until you have seizure offset. When we plot it in the state space, this is what it looks like. Essentially, that's what you saw earlier, the spiral rest state, onset, fast discharges, offset, and back. What we use as data features for the inference is actually the envelope curve here. Here you see it also in this frequency time domain. This seizure here to the top left, fast discharges, and then onset fast discharges. And then something happens, actually the spike wave complexes towards the end that you see here at the seizure end arise, provide you with harmonics. But when you just look at the envelope, it goes down, up, and down. This is what it looks like. It corresponds to a projection in two-dimensional space, in this two-dimensional plane, this being the resting state, and this being the seizure state, the ictal state, and these are transitions. So these are real data here. Yeah, this is what we use as a data feature, and then we put it into a Bayesian inference framework where we estimate a posterior probability density for the parameters at stake. And in our case, it's the epileptogenicity. So it's the excitability parameter in this model that we discussed. So it's one of these parameters. This is in the codimension 3 model. This is the excitability. In blue, you see real experimental data, so sampled from a patient's seizure. Please note it's for one brain area. Note it's multimodal. Yeah. So it's multimodal. Yeah. So With taking an HMC approach, in this particular case, Hamiltonian Monte Carlo, we can actually sample this distribution. What we do very often is that we look for the center of mass of probability, and we estimate this. If we take just a simplistic second order approach where actually the distribution is approximated by a Gaussian, this is what you get in red, standard variational inference. It gets you nowhere essentially, because you remember that this is a target for surgery, and you need to know whether you're here or whether you're here, or whether you have the two options. And then it's a clinician that needs to decide that. This for one brain region. When we span it in two brain regions, this is a type of landscape that you get with multiple nodes, where we have to make decisions whether we deem an area epileptogenic or not. So this is a challenge. Then we communicate the information to our clinician collaborators, and the patient is being operated. And here you see an example, post-surgical MRI, that you see here, overlaid with what we estimate as being a heat map. So essentially this would correspond to the green cross that you see here, overlaying it with the post-surgical MRI. Here was very good correspondence. And the way we communicate it is to the clinicians is we build a clinical report, which we build across brain regions, where we are providing the posterior probability distribution for each brain regions, and we score each brain region, and that was actually their wish, that we place a threshold in which we identify it as epileptogenic or non-epileptogenic, but still provide all the information so they can actually judge for themselves in terms of multimodality and the confidence that we have. We derived a set of confidence metrics quantifying our certainty that we have about the inference. And this is an example, this is the example I just showed you, where there was a good overlap with the surgery that was performed. And here you see the heat map where there was a bad overlap corresponding to the surgery that was performed. So this overlap is our outcome quantifier when we compare it, when we perform statistics. There are different ways of doing it, but that's the best or most informative one in our hands. Here you see for a cohort of 54 patients where we actually compared the prediction that came from the virtual brain twin against the clinical hypothesis. So what they based on the traditional ways, how the clinicians have estimated that. This is precision, which is actually pretty good, 77%. And then it drops down for not seizure-free patients, yeah, versus seizure-free patients. It drops down to 62 when we compare it against clinical hypothesis. And for the patients where we had post-surgical MRI available and could actually overlay it with post-surgical MRI, it drops even much more. This is on the right-hand side, not seizure-free, on the left-hand side, seizure-free, and it drops significantly, not for the clinical hypothesis, but for the predictions coming from VEP, virtual epileptic patient. This is the acronym we use for the workflow using the virtual brain twin for in epilepsy applications. Yeah? So this has been taken as a motivation to do this more systematically. Here, quick information about the performance in terms of low resolution and high resolution models. Remember here, this is on the millimeter scale, this is on the multiple centimeter scale, seizure-free, not seizure-free, precision recall, the same situation. And I guide your eye towards these two ellipses. Look at the precision values for the seizure-free patient. And it really starts clustering up on the top level around 1.0 for the seizure-free, the precision value. And that's due to this much improved source to sensor mapping that we have in the high resolution. Yeah? Independent of the prediction of the dynamics, even this is already a huge added value. So coming towards the end, I wanted to show you where is this added value coming from. So I remind you of actually what is happening here. Yeah? Imagine you're a clinician, you have the patient based on the EEG, or so scalp EEG, or whether there is a lesion, yeah, or on the clinical symptoms, you have to make a decision where you make the implantation of the SEEG. Yeah? So you make the implantation, you place the electrodes, yeah, you drill the hole, you insert the telluride needles, and then based on the totality of the information that you have, you make an estimation of the epileptogenic zone, for instance here. Yeah. Yeah. What we do by linking it to the virtual brain twin is we are building a full brain network model, cortical, subcortical areas. Yeah. And then we are performing using in the inference framework the same data that you have here, even though we are informing just this subset of the full brain network. This is called partial sampling. Yeah. And we perform inference. And that allows us actually to make inference about brain areas that have not been measured and potentially fall outside of the visible regime to the clinician. Yeah. And when we do this, this is actually not an exception, but it actually, when you look at this is a false discovery rate. Clinical hypothesis for seizure-free patients, not seizure-free. This is the same cohort I showed you earlier. No distinction between FDR false discovery rate between seizure-free and not seizure-free, but so it's very good for the seizure-free, for the virtual brain twin, and huge. So false discovery rate being small is being good, huge for the not seizure-free. And when you look into all these patients' brain areas, it turns out that a third, 29% of all cases of identified epileptogenic zones that falls into this category here, where actually the identified epileptogenic zones are outside of the accessible area via SEEG. So the added value is the augmentation of the value of the data by actually linking it to knowledge that we have in the model-based domain. This is what we do, and this is where it comes from. And what we have started doing in 2019, we started a clinical trial under the direction of Fabrice Bartholomé in Marseille, multi-site open trial, yeah, where data are being collected from multiple patients in 13 reference centers. They are being centralized. We virtualize them in Marseille. We build the type of record, the type of clinical report I showed you, and we send it back. We send it, in fact, back to half of the centers, so it's randomized, and not to the other half of the centers. And if they take into account this information that is coming from the virtual epileptic patient in their decision-making. Where we stand today is we have run the trial through a very difficult COVID-dominated period for 4 years, and we just finished inclusion in June this year. So we have 356 patients. That was the target. That's what we needed. So we finished patient inclusion. 300, almost 300 have been randomized. We have virtualized 160 thereof. Yeah, 133 were then under judgment in the branch where they are actually being taken into account for the surgeries, and 178 surgeries have been already performed. We have to wait 1 year after the surgery in order to have an estimation whether the patient is seizure-free or not. So we will have the final results of this prospective clinical trial in summer next year. And that's what I wanted to share with you, essentially show you the two different uses of going from the patient brain, building a virtual brain twin. And this is a diagnostic arm that we discussed in much detail with the epileptogenic zone estimation, but we can also use it as a kind of in silico platform for virtual surgery. This is something that we do often in Marseille, or to optimize brain stimulation parameters. Yeah. And as I alluded to earlier, there are loads of efforts at the moment of taking this digital twin cycle and generalizing it. Questions that appear are, what is the best representation mathematically of a digital brain twin model? What are the data features? For seizures, it's clear. Yeah, clear, quotation marks. We can have a hunch, but what are other good summary statistics for other type of data in other diseases, yeah, that are informative for the implementation on this type of level of virtual brain twin, et cetera, et cetera. But this is ongoing activity and a very hot topic of research. Let me stop here. I thank my colleagues, my friends that have contributed to this over the last 20 years, my clinical colleagues and postdocs and students, et cetera, that are working with me and actually also our funders. Thank you for your attention.